Hello. This is Selim Romero again. And today we're gonna go deeper into cell type annotation, which is the hardest part. And after it, the downstream analysis should be easier. And so far, I'm gonna keep using the same data set I've been working in the previous videos. I'm gonna work with this colorectal cancer from human. I double click on it and should appear here. That means that I can do scGEAToolbox, SCE in the parentheses, and the data set should come out. And this is what we have here. Previously, we had these cell types, and we have these clusters IDs before. Too many clusters, so I see like 1, 2, 3, 4, 5, and kind of 6, 7 islands. So I'm gonna try to lower this down. For that, I'm gonna use the latent clustering as before. SCE equals to latent clustering. Then I'm gonna put the object, and the second is gonna be the resolution. Here I wanna use a smaller resolution, which is gonna decrease the number of islands. It might fail the first time this function, but it's just because it's initializing the Python. You have to run it a second time and should come out. I'm gonna stop it until it finishes. Alrighty, it finished and it found 11 clusters. It's a little better. It's not what I wanted, but it's gonna be fine. So we're gonna again scGEAToolbox in parentheses the object. This time we have to load it from the workspace as I am doing now. Otherwise, it's not gonna show the updated clusters. After it, we should be able to see 11 clusters only. And this is what we got. Cool. As I expected, island 10 is isolated, island 11 is isolated, 9 and 6 kinda isolated. And here we have a rich population that is with a lot of heterogeneity, and that's gonna be the hardest part. Again, here, what we're gonna do first is gonna be annotate and do Panglao gene markers. Continue. We let it know that it's human in this case, and just automatically. Gonna stop it and come back. Alrighty, after it finishes, we just merge the clusters, and that's what we get. Something very similar to what we had in the previous videos, just with different resolution, but now we have to verify that those are correct cell types. One way to start analyzing if those are correct is going to literature, internet, whatever, any method, and we can start looking, for example, cell types in large intestine, depending on your sample. So we look for this, and I click on this first, and this is more or less what I expect in my sample. Goblet cells, enterocytes, intestinal epithelial stem cells, enteroendocrine tuft cells, macrophages, B cells, and T cells, dendritic cells. Mostly these cell types, I've seen it in colon tissue, specifically in mice, although we are working with human. This is another one. Here they have goblet cells, TA cells, transit amplifying, enteroendocrine, enterocyte absorptive. Here are already some specialized or sub-cell types of colonocytes. And potentially we are going to find those. The first thing we can do is going back here and start looking for some marker genes that should be pop up in there. The first thing here is that I have some mice markers that we can explore. Those might not translate precisely, but we can explore those. So if we look at the first, let's say EpCAM, it's a very important marker in intestinal samples. So we just do EpCAM, normalize A and B together, and we're gonna get this. Okay, it seems that mostly everything is lighting up EpCAM, and I think it's mostly colonocytes, epithelial enterocytes within this sample, but this in the top are the fibroblasts and the macrophages as we have here. So that means we have to disseminate more of the markers. Now, if we look for example, fibroblasts, let's look at the markers for fibroblast here in this button here. No, again, paste. We're gonna paste the list. We're gonna select and paste this list I selected, and I want to normalize A and B. Col1A1 is a good marker for fibroblasts. So it's lighting up this in this corner, which is yellow here, but not that many. So potentially those are not fibroblasts. But here, in the middle here, it's potentially fibroblast. So if we look at Col3A1, again, this in the middle, so this is definitely fibroblast. Here, maybe not all here in the bottom left. And again, Col6A2 verifies that. So very likely, this selection, let me just brush mark. All this selection is fibroblast potentially. Okay, if we keep looking now, macrophages. Let's look for the macrophages markers, and then we paste that list and normalize. It's gonna bring out this. CD163 is not lighting up. I don't know if I need to research if those are mice or not, but I'm gonna cover in a few minutes that. This one is poorly there, but it's there. Potentially those are macrophages. This other marker is not there, and MRC1 is also not there. So we have to verify what's in there. And what I will say here is that these markers potentially not gonna work for this work. Here's another cool stuff. After you figure out a marker list, what you can do is cell type 1, do a tab and put, for example, EpCAM, and for example, MAC2. MAC2 is a marker for goblet cells, and those are also within colonocytes as well. Those are not part of the immune system or the smooth muscle cells. So this can be used to separate, for example, colonocytes. And if we utilize this, we can utilize for customized annotation. We can come here in annotate and then use the second row using customized marker genes. You select no to load SE type, and here we just paste our customized markers and paste, and we can, oh my bad, there is something we have to do first. We have to select first over what we want to assess the markers. So we gotta do it over the clusters because it's gonna score that community only. Then we select again and copy, and then we annotate, customize, no, and then do it again and just automatically. There we go. Interestingly, the colonocytes here are as I expected. Here, these are with an O, and these are fibroblasts. Before, here in the top, we had macrophages. We are not getting it right now. So here, according to what I see, I think we should take different venue to annotate it. So one way is again going to cluster ID, and we are gonna do a good way to find what's particularly in every single cluster and what genes makes it unique per se. So what we are gonna do is execute here. First we can do this, find markers. which is this, the marker heat map, but only select find all markers, and you're gonna do for cluster ID. It's gonna take time, so I'm gonna stop the video for a few minutes. Okie dokie, after I finish, the loading is gonna go away and we will have this table viewer. What we're gonna do is export as Excel here, and then I'm gonna call it all markers, and that's gonna be enough. And we can close this. It takes time, so it's gonna take like 10 minutes, could take more, could take a little less time. And what I'm gonna do is open that, and here we are going to have gene markers for every cluster ID. For example, for the first, the top with the largest, with the more confident p-value and largest fold change, it seems, not exactly sorted by fold change, is gonna have the information for whatever gene marker, what gene markers a cluster may have. So what I'm gonna do here now, I have a function that processes that table and sorts it by log fold change and keeps only the confident top 10, 20 depending. So here I'm gonna ask it only the 10 top, and it's gonna open this table. Wait me a second. I think a marker, no markers. So like this. And the T should be loaded. Okay, it seems like I have everything, and I'm just gonna run whatever I have here in my script. I'll pass it to you. I'm gonna have it in the GitHub, so all of you have access to that. And I have now a new extracted markers spreadsheet, which gonna have the top 10 for every cluster. These are the top 10 for the first cluster. So here we're gonna do something very interesting. One thing first, it's gonna be bringing this, the UMAP. Second, it's gonna be having this in the corner right here. And the third is gonna be having this website. And we are gonna do a few things here. Again, our website enricher, and we are gonna start asking enricher not only Panglau. Panglau is also here in this database, but we're gonna feed the top 10 markers and just submit. And we are gonna look for cell types. And here it's suggesting is cell marker is enterocyte, large intestine, exactly what we expected. Oh, sorry, I should have made it smaller. Right, like this. So the cluster 1 is enterocyte, epithelial, all those are same. Here's pointing out to epithelial. This is saying monocyte, but monocyte, I don't think it is. The augmented cell marker is telling it is large intestine enterocyte. So everything is pointing that this cluster number 1 is enterocyte. So if we do the same, but for the second, which is just aside that, we grab this other set and paste it, and we submit, and then we go to cell types. This is now a little different. It's coming different. Epithelial cells in Panglau augmented. I believe it is. Colon tissue. It's too low. We usually have to look only at the top. Cortex, stromal cell, lung mouse is not that. Stem cell, lung human. It's not that far. This is telling large intestine. We're gonna look for if it is epithelial or not. So it's telling that it's potentially something else. colorectal carcinoma is not significant, but is possible. Epithelial cells. Okay, and then we can go back and forth looking at all the clusters and see what the signature genes are. We go for this, enter, to submit, cell types. This says something else, stem cell colon human. That's something I would like to see, because I know here we have some stem cells. Here, for example, I'm interested in that type of cell markers. We can start building our cell markers, not only for the global, but also for the subtypes. And for example, I can just call this stem, and that stem is gonna have the markers potentially that are lighting up here. Those are here. I want to see what's in there. Stem cell SMOC2. It tells SMOC2. That's something, but probably we gotta look deeper into what other type of stem cell could be in there. But like this, we can start building the markers. So I'm gonna go back to cell types and see if something cool. Enteroendocrine, that's possible. What the markers are. Okay, that LRRTM4. Stem cell pancreas, we are not interested in that. So we just roll back here and we start looking around if something makes sense to what we saw in the picture diagram. Well, so far, still I believe everything here is colonocytes, but we have to still keep looking at. Let's look at something different at this point. If we come back here, I want to look not at the I can look at the fourth cluster, but I think it's gonna point out something similar to be intestine related, stem or colonocytes. I'm not really worried about that. The problem is that find markers compares the cluster, for example, one against the rest. So it already loses resolution if we are not packing all this big island together. That's why we are getting some mixtures of genes of different signatures. For example, this is myofibroblast. I don't know if that is valid here. Very likely not. But it's possible that this goblet cell progenitor in large intestine, those are part of the colonocytes. That cluster number 4 potentially is goblet cells. And so far, so good. Sorry, we can go back. I'm gonna enrich it again. And if I am interested into that cell type, I'm gonna go back and look at the markers pointing out that. But what was that? Progenitor goblet cell. Going to the table and GLNTS that gene. I can try to write it down. Goblet tab and then what the name was. Let's grab the name. Large intestine. Interesting. It's this and this other, the two of them. So it can be cap N8. Cap N8. And here's something I want to bring out again. We don't know if that's really what we should. Secretory progenitor, that's also really cool. But it's esophagus. We gotta be careful what we grab from here. Epithelial progenitor cell, but from lung. So the same markers in different tissues matter. It's gonna tell different cell types. So what I'm gonna do, for example, this is part of what you can look here that simplifies a little bit things, but there is another way. So this way it's complicated because you have a lot. It's easier if all of them convey the same similar cell type, but if not, we can do something simpler than this. We can go and look for PanglauDB database, Panglao database website, which is gonna be this PanglauDB. And here it's cool because if we look in, we're going to search, here we can copy genes. We can, for example, copy, let's look something. I wanna look again at the first cluster. So if I paste that and tell it, oh, it's human. Include tumor cancer samples, yes, because in this case we have it. Include non-adult and non-primary samples, embryonic, fetal. In this case, no, because the cell lines were retrieved from donors that were about 21 years old. So we are gonna grab this. And let's see, nothing came out. Not surprised, I haven't seen that guy before. But we can go back and forth looking for information of each gene, and can be even not simpler, but oh, this one points out something. Germ cells. That doesn't make sense in my sample. So again, I can go and look for, maybe this one might make sense, but still we can grab them in collection and try to put it and search for them. I think we need comma, and so forth. And it's gonna tell us if something is there. There we go. The query contains invalid symbols. So it couldn't figure out with some gene names. For example, it seems that these ones, it didn't like it. Yeah, this kind of symbols don't like. So we can grab, for example, these ones. These other two. This other. This. And let's see what pops up. Okay, now it did something. Comma maybe? Let's look. Let's see if that works. If not, we're gonna reserve to one by one. Okay, yeah, comma. So it says this, this, and this. So most of my genes didn't show anything, but the very little that were in there were pointing these two cell types. Colon, serotogenic neuron cells. So it seems that the top 10 markers wasn't enough for the cluster 1 in this case. So still we gotta go back and forth, and this is because it's because we have all these markers and are being compared within the similar cell types. So the clustering wasn't great for doing that. Zero expression. Okay. Now what we're gonna do is, in beautiful scenario, for example, if we go to 11, should be some good markers in there. 11. If we grab all these gene markers, in theory, C1QB, I think it's macrophage marker. We go to here, enricher, paste that, submit, cell types, paneth, macrophage stomach human. That's pointing out to that. Let's see where elsewhere. Panglao augmented, dendritics, macrophages. Very likely those are macrophages. So again, if we come back here and look at the table, these markers, C1QB, CD84, FCGR38, those are in two into the macrophages. Macrophages unified human. That seems good. Where is that? Here. So C1QB, I know it's a macrophage marker. So if we grab that, for example, oh my bad, it was CD84, the other one. If we grab this marker and tell human and search, it's gonna tell a human, dendritic cell, and macrophage. So it's potentially dendritic cell, macrophage. Those share the same gene lineage. So what we need to do is grab all the markers here and put it here and separate with comma. And search for it. This one didn't find. Okay, so now we have a rich gene set here. Dendritic cells, where are macrophages? Macro. Okay, here's another trick I forgot. Macrophage is quite in the top of them. Germ cells, gamma delta T cells, unknown cells, dendritic cells. So here is another trick I can share. We have to look at the sample tissue ID, colon, inflamed colon. So this is dendritic cell in colon. No, dendritic cell, colon. Everything is pointing to dendritic cells. Dendritic cells. Yeah, and unknown here, but just don't pay attention to that. I think this is the main lineage of these type of cell types. Smooth muscles, also not that far, I guess. But it depends how clean the data it is. This is in the T cells. Now, we have a grasp of what that cluster is. So I will say we can come back and, for example, look at the customized. I have a set for classical dendritic cells. So we can look at the expression of those. And is it coming? It didn't work. Let's do it again. All right. All this is complicated, I know. We'll look now at the cluster 10 called 6A3. That's definitely fibroblast, is one of our markers. So 10 is fibroblast. Another question we had is 9 fibroblast. We gotta look at this and see what's in there. So we can grab these markers. This doesn't look like fibroblast to me. So we can go to Enricher and look quickly there. Cell types, dendritic cells, the number 9 it says. Let's look for something that says colon or the Panglau. Endothelial cells. Potentially, it says B cells here. But I know those are not B cells because the B cell markers I had weren't expressing that much. For example, I want to show you how good or bad are the markers I had before. So if we come here, if I paste these two and bring it here, I think it should understand that it has to uppercase it. Right, CD19 and MS4A1, it's B cell. So it's not that. Those two we looked before and those are weakly expressed. And if we look for classical dendritic cells markers. I think those are also good markers for dendritic cells. Yeah. And since we use those for, let's look for colon tissue not here. Very few. But again, if we verify our marker set here, it's gonna tell, oh, it's already good or not. This one, it tells is dendritic, but I know those are macrophages. And the dendritic cells can be separated further with these classical dendritic cells markers. Still fine. We can, for example, now look at this set for fibroblasts. Look for it. And fibroblast. So it's definitely fibroblast marker set. And this is for verifying what we see in the differential expression. So what it seems to be happening in this sample is that it needs to be decontaminated. And that's gonna be for a different lecture. So far, what we learned here is that we can build our own marker genes only if it makes sense from the clusters and from what we can find out in the here in the corresponding websites to carve for the genes. So again, let's do this one more time. But what we are gonna do now, it's instead of using that many clusters, we're gonna look for even lesser clusters. Now I'm gonna look for 0.2 resolution. I'm gonna stop the video for a few minutes. Okie dokie, it finished, and now it found 6 clusters, which I hope it looks better in terms of what I wanna do. Okay, this looks way better than what I had before. Try to unify most of the clusters here. Here again, we are gonna try to annotate with the Panglao markers and see what comes out. Human, yes, automatically. Nice. We get enterocytes here, and I know these neurons are also enterocytes, but those are cancer-related cells, fibroblasts and macrophages. That's what we have. I'm a little bit happier with this, but still not that super happy. So what we are gonna do to finalize here, we're gonna look at the clusters here and find all markers, cluster ID, and do the marker heat map again. Sorry, the marker spreadsheet again. I'm gonna stop it and come back in a few minutes. Okie dokie. Sorry for taking that much time. I know the video is already too lengthy, but it's as difficult as this, doing the annotation. Again, we're gonna export, and I'm gonna override the all markers. Yes, please. Once it's saved, I'm gonna close this, and I'm gonna run again this script that has to be in the same location as the Excel, and it's gonna get me the simplified, per se, markers. Now I have something different than before, and I hope that makes it easier. Oh, wait a minute. This is not fine, because the all markers should be 6 clusters only. This is not right. Let's erase it. Okay, we gotta be sure that whatever we find is the same as we had before. So I know this one, I have to trash it away. So again, we look at this and verify that we have the exact clusters we want to work on. Cluster ID. Those are the clusters, and I'm gonna verify again, define markers, until I find just 6 groups in the Excel. I'm gonna stop it and come back again. Okie dokie. Once it's finished again. Here we have the metadata. If I scroll down to the very bottom, it says 6. Great, that's what I wanted. Somehow I didn't overwrite the old marker I had before. So just be careful that you have what you need. This time I'm not gonna close this for God's sakes, and we're gonna go and look at that Excel, and my bad. And perfect, it lights up to the 6th. And now I think it looks a little better in terms of markers. Okay, our task again is gonna be simplifying that. So we are gonna run all this script to extract only the top 10. Sometimes the top 10 is not enough, and we look for the top 20. Now I'm gonna close that one, and I'm gonna open the top 10 extract. And here I'm gonna look at the first set. I'm gonna do this again. So we go here to the website, we put this in the button. There we go. Something like this. And we keep the UMAP in the left, so we have a reference. Okay, if we grab this first set, copy, and go to the enricher website first. And search cell type. We get smooth muscle. Smooth muscle, that's for cluster 1, this big one. It's not finding much stuff here. We're gonna go to I can just put the first one and see if that recognizes it or not. No. What about the second one? This it founded beta alpha. I don't think those are beta cells. So we're gonna go to this other, oh god, this other germ cells, LGR6. Oh, come on. Didn't find that one. Gallant 14, that rings a bell. Let's see. Well, nothing came out. From this cluster, we will have to try to make sense from here. Stem cell lung. It's pointing out to something stem cell related. Basal paneth of epithelium proper. Epithelial sounds correct to me. No. So here we didn't get much. What this means is that we have to increase the search to 20 marker. So I'm gonna run the script again and do it for 20 markers now. Run again, run it again, and then open that Excel. And now copy the top 20. Those gonna be top 20 for that cluster. And let's hope now it got cleaner. Cell types. No, not at all. Intestine stem cell, esophagus, colon. Okay, we got more or less what we were looking for. Stem cell. Intestinal crypt. Yeah, perfect. This is part of the problem. Sometimes the top needs to be even deeper. Intestinal stem cell, Mark 1 and PBX1. Mark 1. So Mark 1 and PB. right? Come on. PBX1. PBX1. So what we can do is go to the tool again and look at the expression, paste. Okay, yeah, A and B, and I hope it only or mostly lights up in the number 1. And yeah, it potentially lights up in all this, which is exactly what I expected. These are colonocytes. But stem cells, I am not so sure all of them are stem cells, but mostly within the number 1. Okay. Again, we can go now for the second cluster, which is going to be this in the left. And we grab all those gene set. And let's ask enricher again. Cell types. Goblet cell. Okay, that makes sense. Goblet cells. Stromal cell, spleen. Goblet cell, large intestine. It is. Oh, I lost it. Okay, it's pointing out that number 2 is goblet cells. Fibroblast lung, that's not it. So we can come back here. Capn 8 and MLH. What I can do is export it because I'm feeling lazy right now to write it down. Open it and just copy the one I wanted to bring up. Goblet cell, large intestine, and it's these 2 genes. I'm gonna copy and I'm gonna bring it here. And that should light up in that section. Okay. My bad. I'm gonna do this. Paste. Copy that. Yes, normalize A and B. And it's gonna light up this. Perfect. I think that's correct. That's goblet cells. This in the right. Those are goblet cells. Okay. One last proof. Goblet cells are MAC2 positive. MAC2. MAC2. Let's see if that also lights up in that yellow section. And yeah, it is lighting up there. That's very likely goblet cells. Here, I'm just going to include into the markers MAC2. MAC2. No, I'll just keep it like that. Sure. If you look at the third cluster, so cluster ID here. Third is going to be the yellow. You can come back here, paste it, and look for it. Cell types. Neural progenitor. MKI67 progenitor cell, large intestine. That's what we're looking for there. It's MKI67 progenitor. So let's look at MKI67 gene expression, and let's include no, that's fine. Let's look for it. And well, it's over the place, but it's mostly populated here in the bottom where this is. So yeah, potentially that is progenitor cells. So what we can do, it's go cancer cells. That also can be cancer cells. Let's have a little glance at the rest, but I like that. Progenitor cell stomach, great. Everything is pointing out to pluripotent stem cell. That's also possible. But we are going to accept for now um, the progenitor cells. Open it. Progenitor, and the markers there are this. I am curious. Sometimes it's refining the markers. Progenitor. I'm gonna keep it like that for now. But here, something we gotta be sure is that we get exactly what we want to find here. So all this, again, these are subtypes of colonocytes. And that's exactly what I expected to find in there. And here, this fourth very likely also has colonocytes, but we gotta look at it carefully. How I know this is because if we go to the batch, all the cancer are here, and here are just the healthy. So some colonocytes should be here. And as I remember, this blue one was classified as enterocyte before. Let's look at it. Yeah, as enterocyte. So these are healthy enterocytes, and here the neurons are cancer enterocytes. Mostly what we are seeing to some extent. So all this took a lot of time for figuring out. There is only one thing I want to be completely sure, and it is that the 6 macrophages and 5 fibroblasts are properly separated. So we're gonna look for the 5th, and I think it looks like it. 5th. Paste. Go to the cell types, and fibroblast. Cancer-associated fibroblast skin. Possible, but I will not call it like that. I'm looking just for fibroblast here. Great. What markers? COL3A1, PRRX1 FAP, COL6A3A. So that's definitely fibroblast. Those markers we had it before. Cluster 5. Now, let's look at cluster 6. And that, I think it's definitely macrophage or sublineage. Paneth cell, that's possible as well. Macrophage stomach, human. Great. Macrophage skin, mouse. So yeah, I think it's macrophages. Monocytes also have some shared markers with macrophages. Paneth cell. Let's look for another one to see if any other place says macrophages here. Somewhere. Here's saying again, Paneth large intestine. It's potentially Paneth macrophages here in Panglao. So I think it's fine to call it macrophages. What markers? Most of them. Okay, so those are properly annotated. So the short conclusion here is that this is how we build the subcell types, analyzing one by one the clusters, but that needs to be done carefully. To conclude in this video, we verified that macrophages are macrophages and fibroblasts are fibroblasts around. So we got the markers, we look at the markers in some other places, and that's lighting up that it's correct. These enterocytes and the neuron cells here needs to be carefully separated. And I think we need to decontaminate a little bit the data set. So we are gonna do it again, the annotation, but with the contamination, where the main topic is gonna be the contamination. All right, that's it for today, and I hope you enjoy it. See ya.